Well, thank you very much. And I think this meeting is important more than just a meeting, but because it collects people that talks about world issues from many points of view, and that's a unique property. And I'm very proud for being invited to give a talk. Now I'll try to put my can you see?
Yes.
Yes, it works.
Yes.
It works. That's really wonderful. It's like a miracle. So coronavirus is a cunning paradise. Actually, all the paradises are cunning. And this, like other parasites, is using the human system to translate its genetic code. But I will not talk only about this, because I think that there is another very acute problem, which is antibiotic resistance, which was described as by the World Health Association as a disaster. So you saw already viruses from all types. I just want to show this is the one that we have now. But viruses can have all types of shapes and sizes. This is the longest, there is the smallest. All are parasites. All take over the cellular, the host machinery. No matter how large they are, the remarkable diversity exists in shape, but not in concept. So you have seen already the virus. I just want to highlight again. There is a membrane, there are spikes, and the genetic code here is on RNA, not DNA, RNA chain that is single chain and positive going for whoever is interested in this detail. So actually, this is what we just saw, and let's see what happens in this gene. So the gene is very long. It's a very, very long one. It is being translated, being expressed in the host cell, and then being cleaved to different other proteins. 1, 2, 3, and 4, I will show it to you in a bit more detail. So the one here, which is a protease number 1, which can cleave more and more. The second one, then there is a polymerase that is single strand polymerase that can polymerase the genetic code of the sort of child of the virus of the next generation, and a spike, and many, many more that are called non-structures. And I don't think we really want to talk all about them. The spike was mentioned earlier, but before I go into it, there is a very nice drawing that shows how all these little proteins are part of this one. And they are being cleaved, each of them, to do what it needs. Most of them are unstructured. No, NS means non-structure. About the spike. So the spike binds to a receptor, to a human receptor that is called ACE2. The name is less important. It's shown here in green. This is the receptor. And this is the structure of the spike in many bacteria, most of them SARS, but have a look how many. And they were examined, their sequence and structure were examined since 2002. It means almost 20 years, until 2019 last year. This is the one that is now. And they vary. You can see here in the table, variations, biggest variations are here and here. It means if we look at the structure in detail in this part, that the part of the receptor that the virus binds. This is the part, and then they vary in many places, but the highest variations are here. So essentially, to try to block the virus entrance to its receptor, it's very tricky. It doesn't always work, and of course, just to copy from another one does not apply here. So they're equipped also with an array of mechanisms to counteract host cell rejections. And also a very clever, as I said earlier, but now I stress it again, very clever strategy to take over cellular processes. And I will talk about the most important cellular process, which is the genetic code translation, because they have to translate their own code. So in normal cells, in human or in bacteria, the genetic code is stored in DNA. These are the letters of the code within the double helix. They are not available here because they are covered by the structure. So the first step in normal cells is transcription to a copy, which is not on DNA, but on RNA, the same material that the COVE has as its own genetic code. And here, RNA can exist as a single strand, so the letters are exposed and can be copied or translated by cellular compounds like enzymes. And then it is being translated by ribosomes which are shown here, to growing cells. I will talk about control in a minute. So all ribosomes perform their major function like decoding and peptide bond formation in the same way. In vivo, they work very fast. They can make 40 bonds between the amino acids, which are the components of the protein, in 1 second, hardly make mistake. Mistake rate is between 1 to 500,000 to 1 to 1 million, is very low. I was a very fast student, the fastest in my class, because I was very poor and I had many jobs and I had to finish my lab very fast. I needed 6 hours for a single bond, and they can make 40 in 1 second. So the ribosome, we like to describe it as a factory that gets the information, the commands from the genetic code, here the RNA that goes into the top floor of our factory, and tracks that are also RNA chains, which are tRNA, that bring the amino acid. And when there will be here the triplet that codes for this amino acid, it will go in and connect to the newly grown protein that comes out from the lower floor. Also, this whole attachment is done in the lower floor. Then the track RNA, tRNA, will be free, will go out. The messenger will progress in 3 letters. It means in 1 code. The protein will be a little bit larger. It means 1 amino acid more. And it costs little energy, not much. It's very efficient. That's all very clever. So what I just said, top floor is actually the small ribosomal subunit, and bottom floor is the large ribosomal subunit. They are connected through the track, which is the tRNA. Here there is decoding in the small one and peptide bond in the large one. This is what happens. So when we got the structures, which took a long time and a lot of work, but immediately we understood how the ribosomes function. And I hope I can show it. And we made a little video for that, which worked half an hour ago. Well, I hope it works. Oh God, sorry. Do you see this one too?
You see the small panel.
You see a small panel? Okay.
Yeah.
But maybe if you try the arrow in the earlier mode, there was an arrow on the lower left.
Okay, but that's not the one I wanted to show you. It's too late now, it doesn't matter. I have one that works, and that's what's important, right? So this is how the ribosomes work normally. And the virus is clever to take over this. So what you see here is the small ribosomal subunit and the genetic code coming in, the RNA, into a path. And that's very important, this path. It goes in, it's all controlled by the blue thing, which is an initiation factor. It means if there is no initiation, there is no a connection between the messenger, the code, and the small subunit. Once this is okay, the first carrier comes, the first track, on another initiation, number 2 in bacteria. When everything is fine, the large subunit can associate with the small one by making bridges, and the active ribosome have been made. So now it works. More and more amino acids are being brought by their tracks with elongation factors. And the ribosome helps in, out, 40 times a second. And the code is being read in the small one, which is the lower part of this body. And the bonds are being made on the large one, and the newly bound protein goes directly into a tunnel. We took away the large one. You can see how it goes into a tunnel, and in a minute, you will see the whole ribosome again, like now. And you can see that the protein starts to come out, now long enough, and it can fold to its structure. And the whole process continues until there is a stop codon. It means that everything is fine. And then a factor, which is called release factor, this gold thing from the left, comes in. It releases and finishes the production. So the protein can come out now, come out of the ribosome, go to where it needs, and all the other components are ready for the next job. That's all. Not the most difficult, and very elegant in my opinion. And the antibiotics, and the viruses take this over. So actually what you saw are two subunits, small and large. They are made of many components, mostly RNA, ribosomal RNA, the green, and many proteins in many colors. And just to remind you, the decoding starts on one side and peptide bond formation the other side. The viruses take this process over. They have a way to manipulate the cells. And it was shown by Thoms et al. Just a few months ago that one of the non-structured protein that is made by the virus can block the entrance of the messenger RNA. Sits here. This is now clear. It can block. To me, it's not clear how the virus code can enter if it's blocked. So I still really don't understand how the virus is so clever. Anyway, this can be stopped by many small RNAs. I won't talk about it now. They can control it. If we want to control the taking over, but we can stop the small RNAs, but we still do not know how to stop the virus taking over. So the other problem that connects to life, to genetic code translation on bacteria is resistance to antibiotics. And I think that the fact that you have problems about the virus, They are also connected to the antibiotics because many patients, as far as I know, many patients develop diseases, bacterial diseases that are caused by bacteria, pathogenic bacteria that is highly resistant. So as far as I know, many of the patients of the virus, the coronavirus, either become very ill or even die. I even know some personally because of resistance. So antibiotic resistance, maybe now I'll be able to show it. Antibiotic resistance is, first of all, how antibiotics work. No, not good. How antibiotics work is that they bind to the active sites of the ribosome. All of them. Hopefully I can show it here. They bind to the active sites, very clever. And we have to remember that they were designed by bacteria in the microorganisms fight, bacteria against bacteria. So here is a very small one called Ada, found in Poland. Look how clever, just doesn't let the code go in. Another one that is very clever is called tetracycline. And it goes into the position of the second tRNA, just occupies the position, that's all. And that's enough. There will not be reaction. The third one is the first to be used by after penicillin as antibiotic, and it's called the erythromycin, and it blocks the tunnel just next to where the peptide bond is being made. That's it. And the last one is the cleverest chemically. It goes into the bond, into the position where the bond should have been. Very, very clever. Look at it. Here the bond should be made and chop, clindamycin, the name is. So the problem with antibiotics, antibiotic resistance is extremely problematic. What I showed until now is where the antibiotic bind, and what I'll tell you is the companies, they only made better antibiotics from those that the bacteria made, but they don't put, they put hardly any effort in making new ones. So the World Health Association thinks that we are soon going into post-antibiotic era. And consequently, the World Bank estimated that almost 4% of the global economy will be lost by 2050 because of resistance. Yet, only very few antibiotics are being in development by companies. Most large companies stopped attempts because there is a huge mismatch because the cost and the human values, which is our life essentially. So you can see it now in two tables. On the left, how fast the resistance grew between 1990 to 2002. It means just 12 years for 3 different antibiotics. And here you can see between 1980 and 2018, how little is the number of new antibiotics, actually hardly any. Not improved, by new. So in order to design pathogenic specific antibiotics, it means one of the problems that increased resistance is the use of wide range antibiotics. We want to design specific ones, therefore we determine the structures of some ribosomes from some antibiotics. We learn many things. We learned that all bacterial ribosomes are similar, but not exact. And in lesson number 4, which I'll show you, not all the other before, I want to show you differences between the large subunit of a non-pathogenic bacterium, that's gray, and the little pieces here in the cyan, which are additions of this particular but pathogen, which is called Staph aureus. So if we focus on them, we can see that the pathogen has little pieces that have structure and are available on the surface, far away from all functional sites. And we think that this can be potential binding sites. Actually, there are many of those that can be potential binding sites. And we tried to make antibiotics, it means compounds that bind there, and 16 of them stop ribosome action. It means there is a hope to find new antibiotics. They can be also environmental and ecological useful, and I want to show this. Almost all known antibiotics have cores like this, which are not digestible and not biodegradable. It means when they go into the ground with the sewage, they can penetrate into agricultural irrigation systems because they are too small to be caught by the purification systems and cause more resistance. And for instance, in 2017, the United Nations, you can see the references here, warned that the antibiotics in the environment was driving bacterial evolution, and it's a very difficult, very big global disaster. So with this, I want to finish except for showing you my blue dream. So this is the life expectancy in the world in 2008. The longest life are the dark blue. This is over 80 years old. And I don't want to go color by color, but I want to show you that there are still places in the world that life expectancy, average life expectancy is about 45 years. And my dream is a blue dream, that the whole world becomes blue. And for this dream, for materializing it, a serious committee is needed by the pharma companies. I hope at one point I'll be able to convince them. So before I go, I just want to thank the Weizmann Institute that let me work. And the person that made the decision is Michael Seller, who is a member of this academy. I won't go person by person, just to mention Professor Wittmann from Max Planck in Berlin, that all what he wanted to understand is how ribosomes work, but he died 10 years before we got the structure. And I want to show you the group that I had in Hamburg and the group that I have in Israel. I want to tell you that in my group, ribosomes are considered sweet. Here is a cake that one of the people did. And I want to tell you that my granddaughter gave me a prize because I'm such a good grandmother. She gave me a real nice prize 13 years ago before the Nobel. And it's more important for me. She called me the grandma of the year, is Ada Yonath. And when I asked her which year, she was at that time 9 years old or something. Which year? She said, every year you have to prove yourself, otherwise I take it off the wall. I want to show you it's still on the wall. I want to tell you that in Israel, ribosomes became popular. Here, it's a carnival, they are dancing, he is me, and she is a ribosome, also young children. And Michel Kishka, who is one of our best artists and writers, drew this and said, this is head full of ribosomes. And if you look, small subunit, large subunit, and symmetrical, and he thinks that antibiotics should be here and here and here and here. Thank you.
Thank you very much, Ada.
I'm happy that I made you smile, although the topic is so sad.
Yeah.